All right, so hopefully you are ready. This is our cumulative review for Bio 110. I am going to briefly go through each of the units, some of the major topics, things that you would see on your final review, or sorry, your final exam, as well as on the general education assessment. Your Gen Ed assessment is your attendance quiz for today. If you are an online student, if you are a seated student, you will be taking it in class. It is a 20 question multiple choice test that is a requirement by the state, and it is stuff that you've seen, is stuff that you know. It's covering the major concepts that we've talked about this semester, so it is also a way for you to assess your base understanding and knowledge for the cumulative final exam. A couple of things that we should talk about to make sure that we're on the right track: your cheat sheet. So you are allowed to use a cheat sheet on the final exam. For my classes is one side of one piece of paper, and it must be handwritten. Online classes must show the front and back in their Respondus environmental check, and there will be a space on Canvas for you to upload your document, just like you saw on the Unit 5 exam for those charts. You can and you should take a picture of the front and back, ideally with something like Cam Scanner that lets you upload it as a PDF. JPEGs are okay, but sometimes they take pictures and they make them really small, and I want to be able to read what you write on your cheat sheet. Seated classes will turn in their provided colored cheat sheets that are available to them. I will be going through them. So one of the things that I want to make very clear is your exam is based on your past unit tests. You can and should be reviewing those unit exams, going back through those online reviews for more detailed information of what is going to be covered, as well as going back through those past tests to look at your answers as compared to my answers, which would be the correct ones. You are not allowed to write any answers from past exams on your cheat sheet. This should be a way for you to reference material similar to what you've used in the past. I will be reading everyone's cheat sheets. If you have any sort of answers that are directly or almost directly from the past exams, you will get a 0 on the final. So just if you are unsure if something should be on or can be on your cheat sheet, either leave it off, edit it into such a way that it is a useful tool and a reference material for you, or ask. Send your instructor an email and ask them. Short notes, no complete paragraphs or sentences, no answers from past tests. Tables are not on the final exam, so tables are fair game. But remember, they're going to take up space. You only get one side of one sheet of paper. My suggestions: review the past tests, take notes on what you've missed and why, and make sure that you can answer them now. If you need to and you want a little support for those sorts of questions, go in and put down some key details. So for instance, if you can't remember the equations for cellular respiration and photosynthesis, put those on your cheat sheet, okay? But don't be writing down like full answers to anything in that way. Add notes to each unit only for things you struggle to remember. You should make multiple versions of a cheat sheet before you get to a final version. And the more that you handwrite these things out, you can write out as much as you need. Make a cheat sheet for each exam, and then go through and pick the things that you need to put onto your final cheat sheet. Okay. Your exam format is 35 questions, 135 points. 30 questions are based on past exams. They might not be exactly the same as what you've seen, but they will be a similar format. Same format as the exam, with fill in the blank ordering, matching, and short answer. You will have 5 comprehensive connection or application questions. So this might be something where I give you a scenario and ask you to explain it in the context of everything you've learned for biology, or it might be a connection question like what we've seen with our connections from the central dogma to evolution. So just be prepared for that. These are going to be things that you might not have a direct answer for. I am asking you to think through the problem and solve it and explain your understanding and knowledge. Everything has to be in your own words. Check your announcements or calendar for date and time, and check with your instructor on when your final exam is. There will be no extensions or makeup exams, and you cannot take it early either. The exam week is December 15th through the 18th, so it will be on one of those days. It should be completed in about 2 hours. Seated classes have an hour and 50 minutes, so that is what the exam is built towards for both seated and online classes. So if you have any particular questions about the cheat sheet or the exam format, definitely check with your instructor. But let's get into the basic concepts. I would like to keep this video in under an hour, so we are going to hit these things. And again, if you have any particular questions, go back through those past unit reviews. Those are all posted for you, or your past lecture videos as needed. So for Unit 1, I know it was a really long time ago, but for Unit 1, we covered 4 chapters. We talked about the characteristics of life and the scientific method. We talked about chemical bonds and the chemistry of water, and that chemistry of water question will be on your final, so do make sure you review that. We went through macromolecules, and we talked about prokaryotic versus eukaryotic cells, as well as the organelles that we find in all of them, and specifically in the eukaryotic cells. For chapter 1, I feel like most of us really got and grasped this and should be good at this because we've covered the scientific method throughout the entire semester. But remember, the steps of the scientific method are observation, and from an observation, you will build a hypothesis in that if-then format. Then you will do an experiment testing your hypothesis with those dependent, independent, and control variables. You'll analyze your results and come to a conclusion whether your hypothesis was supported or not. And then the last step is to communicate your findings, because if you keep them to yourself, they're not useful. That is going to be something that you'll see on the final, as well as probably on the Gen Ed or any sort of things you've seen on labs, as well as this understanding of what's the difference between a hypothesis and a theory. As I think we should understand by now, you as an individual scientist, as an individual student, have a hypothesis. You do not have a theory, because theories are very long-term, large-scale studies with the similar or same results found by many scientists over a very long period of time. So for something to become a theory, it has to be supported in a lot of different aspects. That's why you have a hypothesis. This is one of those sticking points with me and my hubby. He always says, I have a theory, which is colloquial terms, common language for something. But we as humans individually do not have theories. We have hypotheses if we're following the true scientific method. Okay? And then I think for characteristics of life, we've all really gotten a handle on this, and it plays a role in what we determine as our biotic and abiotic factors in ecosystems, which is part of Unit 6. So this ties into Unit 6. Maybe one of those comprehensive questions there. But remember, like, living things must have all 6 of these characteristics, with homeostasis being that ability to regulate inside and outside of the cell. Like, you regulate your temperature and your cells regulate the amount of water coming into and going out of them. You need to be able to respond to stimuli. You need to have biological organization, which does include cells, which is one of the big reasons that viruses are considered non-living, is they are not made of cells. You need to have reproduction. Okay, reproduction can be asexual like mitosis or sexual like meiosis, but the ability to reproduce, another thing that viruses don't really have since they do replication. Evolution is absolutely a key factor of life, and as we know, a big part of Unit 4. And then energy processing or metabolism, which again is the entire purpose of Unit 2. So these characteristics of life really came up time and again through our units, because these are what we base living organisms on, and this is something that we understand in detail through those units, through those concepts we've talked about. So do expect there to be some sort of connection question between the characteristics of life and any of the other units that we've talked about this semester. Okay. Let's refresh chemical bonds. Everyone always, again, you hear chemistry and your brain kind of shuts down. I get it. But remember, chemical bonds play a really big role in that biological organization of life. A lot of people have been like, but how do the cells know to do these things? They don't know. The chemical reactions are driving what's happening in the cell. So when the genes are being expressed, right, it's not that the cell knows, it's not that the cell is consciously making a decision, it's the chemical reactions that occur in there. As such, we need to be aware of the different kinds of chemical bonds we find in our cells. Now, remember, this is all related to the electrons themselves. All of our atoms, all of our elements on the periodic table of elements want to fulfill the octet rule of having that outermost valence shell full, because when it's full, it's happy. Just like you, when you're full, you're happy, okay? So When we're talking about these, this is the building blocks of life. This is what makes up the macromolecules, which then in turn make up the organelles, the tissues, into that diversity of life, into the whole ecosystems, and playing a role within that entire biogeochemical cycle. Okay, so there are three kinds of main bonds that we've talked about: your non-polar covalent, your polar covalent, and your ionic bonds. If you remember, non-polar covalent bonds are not charged. Okay, non being not, polar, think about those opposites. So non-polar covalent bonds share electrons equally. Remember, co means together or shared, and valent is referring to the electrons. So these are going to be things like oxygen, O2, or nitrogen, N2. And that's important because they are less reactive and they are hydrophobic. Generally speaking, your nonpolar elements are hydrophobic, and that would be why lipids do not interact with water, because they are nonpolar. Okay, polar covalent bonds do share electrons still because they are a covalent bond, but they share those electrons unequally, and it results in a charge. One of the nuclei of the atom is going to pull the electrons closer to itself with its protons, and that is going to make that atom slightly negative and the other one slightly positive. Water is a fantastic example of this because oxygens are larger than hydrogens. Remember, this would be like Dwayne "The Rock" Johnson playing tug of war with Kevin Hart. Kevin Hart is not giving up, but he is not going to be able to pull the rope as close to himself as The Rock will. Okay, these are often hydrophilic and they dissolve into their elements, and they're going to interact with water in different ways. They might pick up the hydrogen molecules from the water. There's other things that happen there. Ionic bonds are the give a penny, take a penny, right? We give an electron, we take an electron. In this case, we are changing the entire structure of that element because that element is now either gaining an electron and becoming overall a negative ion, or losing an electron and overall becoming a positive ion. In this way, these are hydrophilic, just like our polar covalent bonds, but they interact with water by dissolving into ions. The water is going to surround those particularly charged molecules, those particles that are charged, and they are going to then be broken apart. They are not sharing electrons, and so they are not going to try to share electrons with anything else. They are literally split apart, like if you were to take apart two different magnets. This does bring us into water, and if you remember, water is a polar covalent molecule. This means that when I look at a single water molecule, my oxygen is going to be slightly negative because it is pulling those electrons closer to itself, and my hydrogens are going to be slightly positive. Remember, if I were to look for those electrons, they're going to be in this solid line here between the hydrogen and the oxygen. They're sharing the electron, but it is going to be pulled closer to the oxygen, making that oxygen slightly negative. The hydrogens then are slightly positive. Because of these charges that we see within a single water molecule, when we have many water molecules together, we also see these dotted lines, these hydrogen bonds, which are the weak attractions between the charged molecules. These hydrogen bonds are responsible for dissolving ionic bonds, for reacting with other polar covalent molecules, and for these major properties of water. There are a lot of different properties of water that we talk about. I want you to be aware of cohesion and adhesion. Okay, remember, co means shared. Ad means it's like adhesive, like your glue or tape. So cohesion is how water molecules stick to each other, and adhesion is how water molecules stick to something else. So a water droplet is demonstrating cohesion because those water molecules are sticking together into a water droplet, and the water droplet sticking to your skin is an example of adhesion because that hydrogen bond is going to be attracted to your skin, which is going to have a slight charge. That is also how we get the solvency, that universal solvent property of water, where it can dissolve those polar covalent molecules and ionic molecules. It has interactions with them. There's charges. So for instance, when salt is put into water, this negative chloride is attracted to the positive hydrogens and is then separated from the positive sodium. In addition, water plays a really big role in temperature regulation because the hydrogen bonds break and form quickly, and they take a lot of energy to break completely. And so water is very stable with that temperature. It's also why ice floats because of how those hydrogen bonds interact. So hydrogen bonds play a really important role in the properties of water. You will have a question on the exam. If you remember in the Unit 2 exam, I gave you this picture. It's this picture, and I ask you some of these questions. If you are unfamiliar or you need to go through those again, definitely do so. Go watch that Unit 1 review, go watch the Unit 1 lecture video, and go look at that Unit 1 exam as well. Okay? Macromolecules. There's no table on the final, so you could put that on a cheat sheet if you wanted to. There will likely be connection and application based questions. Like, for instance, if you need short-term energy, which is the food you are most likely to get it from, and why, and how does that play a role in cellular respiration? Like, we know this stuff. We've talked about it. Hopefully your brain went carbohydrates, because if you want some short-term energy, you eat some carbohydrates. That makes that glucose that is then utilized by cellular respiration. So hopefully that's where your brain went. If it didn't, again, this is just a quick overview. Make sure that you go back and review those other past materials available to you as well. If you remember, though, we did talk about monomers and polymers here. Your monomers are going to be your single units of those macromolecules. Your polymers are the multiple units of those macromolecules. And we are going to have bonds being formed in there through dehydration synthesis. Monomers losing water to build polymers is dehydration synthesis, and polymers gaining water to break into monomers is that hydrolysis. If you remember too, we talked about some of the specific examples of these. So carbohydrates, remember, have carbon, hydrogen, and oxygen, and they are forming a polar covalent molecule. We see polar covalent molecules here. It's why your bread or your pasta soaks into water and absorbs it. We have monosaccharides, which are the single sugars. That's your glucose. That's used heavily in photosynthesis and cellular respiration, or it's built in photosynthesis and used in cellular respiration. Various examples of our carbohydrates. We have the polysaccharides like pasta with those starches, as well as like chitin, what we find in our fungal cell walls. Hopefully you remember that. And in addition, things like cellulose, which is in that plant cell wall. Lipids, okay, remember lipids are your saturated and unsaturated fats, your hormones. Because they're so variable, we don't have any true monomers or polymers, but they are nonpolar and hydrophobic. It's why oil and water doesn't mix. If you remember as well, this is an important component of your cell membrane, and that cell membrane plays a really big role in homeostasis and regulation of water and molecules moving across it, and part of that is because of the nonpolar property of those phospholipid bilayers. We'll talk about that a little bit in the next slide as well. Proteins, we had a whole chapter on protein synthesis and the importance of proteins. Remember, those are going to be your monomers of amino acids and your polymers of the polypeptide held together by peptide bonds. We talked about this extensively in protein synthesis. I'll briefly touch on it again here, but make sure that you're going through it again. In addition, you're going to see those enzymes playing a really huge role. Proteins are basically your phenotype, so make sure that you can pull that together as well. Your nucleic acids, again, are going to be your nucleotide monomers, your nucleic acid polymer, and that is your ATP, your DNA, and your RNA, and hopefully you remember the importance of all three of those molecules. I do have connections written in here in this particular table, so if you're studying and you're wondering how to make the connections, these are some of those major topics to review in order to make those connections. Let's go through cells and organelles. If you remember, we have prokaryotic and eukaryotic cells. Prokaryotic cells would be our bacteria and archaea. Eukaryotic cells are animals, plants, fungi, and protists. It doesn't matter, though, if you are a prokaryotic or eukaryotic cell. All cells have genetic material, DNA ribosomes that allow us to make those proteins. All cells have a cell membrane and all cells have cytoplasm. Again, this is one of those key characteristics of life, having these cellular structures, specifically with that genetic material, cell membrane and cytoplasm. Those are going to be incredibly important because, again, your cell membrane, for instance, is that barrier that is providing that homeostasis, allowing things to move across it effectively. And it's like a separation between the inside and outside of the cell. The cytoplasm holds all of the organelles. In the eukaryotic cells, it holds the nucleic acids, so that nucleoid region, as well as ribosomes in our prokaryotic cells. In addition, you should be able to match the organelles to what they do. So if you can't remember that, please go review Unit 1. We do have a lot of similar organelles between plant and animal cells. Remember that plant and animal cells both have mitochondria because cellular respiration is required by all eukaryotic cells. They have the endoplasmic reticulum to help them build lipids and proteins. All cells have ribosomes because without ribosomes, we cannot do protein synthesis. All cells have the Golgi apparatus when we're talking about our eukaryotic cells, as well as the nucleus. Remember, separating that DNA from everything else. And we do talk about the nucleus and the DNA more in detail in mitosis, meiosis, and protein synthesis. Matching for these organelles, either whether they're prokaryotic, eukaryotic, or both, or if they are plant cell or animal cells, or what their function is. Those are very likely to show up on your final exam. Okay, that is Unit 1. I go over it a little bit more because it's a relatively older concept. Let's go into Unit 2. So for Unit 2, we covered chapters 5, 6, and 7, which was basically metabolism because we talked about enzymes, diffusion and osmosis, photosynthesis, and cellular respiration. I know that you are all probably panicking a little bit about photosynthesis and cellular respiration. You don't have to. There are no tables on the exam, the final exam. I have tested you on that. These are going to be more application and recognition of those processes questions on the final. But let's talk about enzymes quick. Okay. Hopefully you remember that enzymes are proteins, and as we've learned, proteins are incredibly essential to life on this planet. Proteins that are enzymes are not used up, and they speed up, they catalyze reactions. They may speed up or make reactions easier by lowering the activation energy required. Remember, if we see these charts with these two nice humps here, we start with the same reactants, we end with the same products. One of these processes takes less energy and is therefore a enzyme facilitated process. In this case, this would be our blue line. Our green line, the one that is much larger, is going to be without enzymes. So let's put that here, right? Minus enzyme, with enzyme. And I do like that analogy where we're talking about building bird boxes, right? And if I can build a bird box with a hammer and nails, that takes a lot more energy because I have to physically pound those nails into the wood. Whereas I could also utilize a nail gun and have the same product and have it be much easier to accomplish, which allows me to accomplish more of those reactions within the same time period. Enzymes are controlled. Enzymes are controlled in a variety of manners, including things like pH, gene expression and regulation, because again, that's where we build our proteins from, and things like competitive and non-competitive inhibitors. Enzymes work in that lock and key where they are substrate specific, and we're going to have particular enzymes for particular jobs because that makes them most efficient. If enzymes are not specific, they are not overly efficient. Competitive inhibition will block the enzymes directly from binding to the active site. Non-competitive inhibition changes the shape of the active site. Okay? Diffusion and osmosis. Remember, diffusion is the movement of solutes and or solvents from high to low concentration gradients, whereas osmosis is specifically the movement of water. We talked about the permeability of the lipid bilayer. It does not let everything through. This is going to maintain the integrity of the inside of the cell, but small nonpolar molecules and small uncharged polar molecules, or molecules that are not charged enough, very easily move across the membrane, which is why we can have osmotic environments that are not the same as our cells, and that can be potentially problematic. If you recall, we have three types of osmotic environments: hypotonic, right, where the cells increase the water quality, the water amount, quantity, not quality. Water runs into the cells or moves into the cells fairly quickly. In a hypotonic environment, animal cells will swell and burst like an overfilled balloon. Plant cells with that cellulose cell wall and that large central vacuole like to be in that hypotonic environment. In an isotonic environment, water moves equally into and out of a cell. This is ideal for animal cells, but plant cells will wilt a little bit. Nobody likes to be in a hypertonic solution because water is leaving those cells and they become dehydrated. And I think we've learned that dehydration is not ideal for any cells. So all cells will shrivel in that environment. Okay. Remember, then we went into ATP. ATP is essential when we talk about ATP. We talk about energy, we talk about fitness. This plays a role in every chapter, every unit we talk about for the rest of the semester. Expect some sort of connection application question of ATP and metabolism to the other concepts we've covered this semester. Remember, carbohydrates are the main source of our energy. Carbon, hydrogen, and oxygen are easily rearranged into carbon dioxide and water, or vice versa, through photosynthesis and cellular respiration. Okay, ATP, energy, fitness. We know this, right? No fitness, no babies. Biology is all about making babies. So remember that photosynthesis is utilizing carbon dioxide and water with solar energy to produce glucose and oxygen. They are not intentionally producing oxygen. That is a byproduct from splitting water molecules in the light-dependent reactions. They are intentionally building glucose, but they are not building it for us. They are building it for themselves. They will store that glucose long-term as starch. We, as consumers, are going to utilize the oxygen produced as well as the glucose produced through cellular respiration to generate ATP. Remember, there is not 100% conversion, and we do lose some energy as heat. And we also then produce water as the end product because we need to attach hydrogen ions to an oxygen to neutralize them. And we produce carbon dioxide as a waste product from splitting those sugar molecules themselves. These are balanced equations, they are balanced reactions, and these processes are going to be opposites of one another. Do remember that plants do both. Animals do cellular respiration. Plants do both because plants have mitochondria as well. And again, plants need energy. Without energy, they cannot grow. They cannot not put out new cells. They cannot do any of the reproduction processes that they need. Okay. Again, the tables are not on the final exam. I'm not asking you to memorize these. If it helps you, put them on your cheat sheet. Remember, the light-dependent reactions happen first. Those occur in the thylakoid membrane when water is split into hydrogen ions and oxygen, with the oxygen being released as a waste product. Then the hydrogens are trapped by NADPH and used to generate ATP, which moves to the stroma, or the space of the chloroplast, for those light-independent reactions, also known as the Calvin cycle. Carbon dioxide is utilized here, and it is rearranged using the energy from ATP and NADPH to build a carbohydrate. Remember, we have to have carbon, hydrogen, and oxygen to build a carbohydrate. We get that from the CO2 and the hydrogens from the water, and then that ATP, you need energy to be stored long term. ATP is not stable, so carbon dioxide added to that ATP is used to rearrange into that glucose molecule. Okay. Hopefully this is ringing some bells. Again, very detailed review in Unit 2. Strongly recommend. There is no table for cellular respiration on the final exam, but if you remember, we do talk about the 3 steps. There is glycolysis that occurs whether or not we are going through aerobic or anaerobic processes. So glycolysis is the breaking of glucose. Glucose is too big to fit in the mitochondria, and so it needs to be broken down, which takes a little energy. It is done outside of the mitochondria in the cytoplasm of the cell for all eukaryotes, and it does not require oxygen. Now, if you have plenty of oxygen because you're just sitting and listening to the lecture video, you are doing aerobic cellular respiration or aerobic respiration, which includes the next two steps of the citric acid cycle and the electron transport chain. Both of those require oxygen because they are in the mitochondria. Remember, you breathe oxygen for your mitochondria. That is going to generate carbon dioxide as a byproduct as well as ATP. The majority of our ATP coming from the electron transport chain and that use of ATP synthase to generate that ATP. Again, hydrogen ions loose make things acidic, and so we need to capture those, and oxygen is that final electron acceptor there. Now, if you were listening to this while you're running a marathon, which cool on, I mean, I guess if you want to listen to me, right? If you're doing an anaerobic process, you will be switching into a fermentation pathway. In humans, this is going to produce lactic acid, so it's lactic acid fermentation, and that's what your muscle cells do when there is not enough oxygen to support aerobic cellular respiration. This is a good thing because this increases your aerobic capacity by telling your cells that you need more mitochondria, which is why you can maybe start running. Maybe you can't run a full mile, but if you stick to it, you eventually will be able to run a full mile without feeling completely winded. I know I couldn't do it now if I wanted to. I haven't because my knee for a very long time. If my knee worked, I would be able to do it longer term. It's why working out gets easier the more you do it. Walking stairs gets easier the more you do it. You are telling your body, I am needing more oxygen, and you build more mitochondria as a response because your cells need mitochondria and your mitochondria need your oxygen. If you are doing that fermentative lactic acid pathway, you are exceeding your aerobic capacity, but your body still needs energy. You are still utilizing your muscles, and so your body is going to switch to that kind of emergency mode to provide you with the energy that you need. If you remember too, we did talk a lot about the relationship between photosynthesis and cellular respiration. This is because as we get into food webs and things in Unit 6, without producers, there are no consumers. Hopefully, we've all built a greater appreciation and understanding of plants and algae and those producers, because without them, consumers eventually die, and not just from the lack of oxygen in the environment, but from the lack of glucose. Plants are so key to our planet surviving. As we start with that solar energy, only plants and other photosynthesizers can utilize that solar energy and produce a form of energy that is able to be used by consumers. These are balanced systems. They are balanced equations. This is absolutely something that is key to understanding life and metabolism. As always, this is a quick review. Go back and rewatch those other lecture videos, those other review videos. Let's get into Unit 3. I know, right? You're like, whoa, Ms. Larson, we're cruising. Yes, I'm going through an entire semester and hopefully a little less, maybe a little over an hour. I'm aiming for an hour and 15 minutes at the most, okay? So, Unit 3, this is cellular reproduction. So this is mitosis, meiosis, and genetics, as well as talking about things like cancer and non-disjunction. Do remember this was a very vocab-heavy unit. You will be asked to recognize those vocabulary words. You will be asked to match those vocabulary words. You will be asked to utilize those vocabulary words. As such, here are the really big important vocabulary words you need to have. If you go through these and you can't define them off the top of your head, flashcards. Put some little notes on the ones you really can't remember. Draw pictures, label pictures. I'm a big fan of drawing and labeling pictures on cheat sheets because then you are not writing answers from the test and you will be able to identify them. A lot of people having a picture helps. I'm not going to go through all of these here. These are things that were listed and available to you in Unit 3. Definitely do some vocab flashcards just to refresh your memory. I am going to talk about a couple of them really quick here because understanding cellular reproduction and genetics, we absolutely need a couple of these to be very fresh in our heads. First, remember that haploid is half the number of chromosomes. Diploid is 2 chromosomes, so you got pairs of chromosomes. Homozygous is going to be when we have the same allele. Heterozygous is when we have different alleles being alternate versions of genes. Your genotype is the genetic part of your code. Your phenotype is the physical expression. Dominant and recessive is usually the genotype, being homozygous dominant or homozygous recessive or heterozygous. Hopefully those are things we remember. And then your phenotype, you would be either dominant or recessive with that physical expression. Ready? Cellular reproduction is going to be mitosis or meiosis, and we did a lot of comparison of these, mitosis being that asexual reproduction where meiosis is sexual. We did talk about how sexual reproduction can be really beneficial because it halves the number of chromosomes, recombines those genes, and creates unique individuals, but asexual reproduction can be very important as well, especially for some organisms who choose to just make copies of themselves and guarantee their genetics in the next generation. For humans, we rely on mitosis for growth and repair, and meiosis for new individuals. Mitosis is going to produce identical cells unless something goes wrong. That's when we get cancer, whereas meiosis is those unique gametes. Okay, hopefully you're remembering a lot of this. If you remember, they all have, both mitosis and meiosis has interphase with that DNA replication, cytokinesis, and we start with the same parent cells. Meiosis is very unique because we have crossing over, independent assortment, and fertilization. These questions are absolutely 100% going to be a comprehensive sort of combination question on this test. You saw it on the Unit 3 exam. Definitely review those. Do not write a full answer for this on that cheat sheet. That will earn you that 0 on that final exam. Let's quickly review mitosis. Remember, mitosis, the goal is nuclear division. Cytokinesis is the division of everything else in the cell. It is its own process. Interphase is going to happen where we will see growth of the cell, duplication of the organelles, as well as duplication DNA synthesis. Once we have good checkpoints and everything is cleared, we will move through prophase, metaphase, anaphase, and telophase in that order, with cytokinesis to follow in most cells. Remember, cytokinesis can be ignored in some cells if that is the function of that cell. So remember, our muscle cells, we want them to be multinucleated. We do not want to have a lot of divisions in our cells. If you don't remember what these phases do, flashcards. Draw the phase, write what it is on the other side, and go back and forth. You might be asked to identify mitosis or meiosis by image like you were on the Unit 3 exam. We can always tell if it's mitosis because we're going to see one set of divisions with 2 cells at the end, and those 2 cells are going to be identical clones to the parent cell. If something goes wrong, if the cell starts dividing out of control, if we have abnormal nuclei, if our cells are not following the checkpoints that are appropriate, that is when we get cancer. Mitosis happens quickly and irregularly, and that is what we call cancer. Once it can invade those other cells, that's when it's the cancer with the big C. Meiosis, on the other hand, is going to produce new individuals if fertilization follows. So meiosis is just to produce the gametes. Gametes are going to have half the number of chromosomes as the parent cell. So in humans, as an example, we have 46 as a diploid organism. Our gametes have 23 chromosomes, 1 from each pair, which is why companies like 23andMe are talking about genetics. The purpose of meiosis is to reduce the chromosome number by half and give us genetic recombination. As such, we have meiosis 1 and meiosis 2, with meiosis 1 resulting in that genetic combination through crossing over in prophase 1, where the homologous pairs find each other and share segments of DNA between non-sister chromatids, vocab here. Okay, metaphase is when the homologous pairs align in the middle independently of one another, and that's where we see that independent assortment shuffling up those chromosome pairs. In anaphase 1, our homologous pairs separate, and in telophase 1, we have cytokinesis because our goal here is 4 new cells always. Unlike with mitosis, we may avoid cytokinesis as a function of the cell. We do not avoid that in meiosis because our goal is 4 haploid gametes. We want 4 cells. At the end of meiosis 1, we have 2 haploid nuclei because as our image here, which I know is small, but you can zoom in, we have 4 chromosomes to start with. At the end of telophase 1 in cytokinesis, we have 2 chromosomes, 1 in each nuclei. Meiosis 2 is similar to mitosis, where we see the division, but we are seeing the division in 2 haploid cells, resulting in our 4 unique gametes. Meiosis can also go wrong. When meiosis goes wrong, it's often through non-disjunction or a failure to separate, where either the homologous pairs or the sister chromatids do not separate, and we get gametes with abnormal number of chromosomes, either n plus 1 or n minus 1. This can happen during anaphase 1 or anaphase 2, with anaphase 1 resulting in 4 abnormal gametes and therefore being more problematic, because if any of those gametes are fertilized, we get a 2n plus 1 or a 2n minus 1 individual, and that is usually not conducive to life. Or in meiosis 2, we have 2 abnormal gametes and 2 normal gametes. If the normal gametes are the ones that are fertilized, we would never know non-disjunction occurred. Okay, hopefully we're remembering that. Let's go through some of the main concepts with genetics. The biggest thing being that, remember, we are looking at genotype and phenotype. We are looking at the genetic combination of alleles in individuals that is going to then give us the physical expression, which plays a role in natural selection, diversity of life, ecosystems, and evolution. So when we look at Punnett squares, for instance, it's a really good way for us to refresh and recap on these. In our Punnett squares, if we cross individuals, we are going to have different genotypes and phenotypes. So I have a Punnett square here where we have a purebred purple flower being crossed with a purebred white flower. This means that in the family history lineage of these purple flowers, they've always been purple, white has always been white. In this case, when we cross these two individuals, all of the offspring are purple. That is the purple phenotype. That is the dominant phenotype. White appears to disappear, but those white individuals are not completely removed. Those alleles are not gone. They are just hidden, which is why they are recessive. If we cross then those two offspring, which would be our F1 generation, so these are our F1 individuals, okay? If I cross those, this is F1, this is F1, okay? Those are two heterozygous individuals. I now get one individual who's homozygous dominant, two individuals who are heterozygous, and one individual who's homozygous recessive. So I end up with a genotypic ratio of 1 to 2 to 1. And I end up with a phenotypic ratio of 3 to 1, where I have 3 individuals who are purple and 1 individual who is white. Remember, though, that if I have a dominant phenotype, I could be either homozygous dominant or heterozygous, because it's that first allele that is expressed, and it's that first allele that determines the phenotypic expression. In order to have a recessive phenotype, I have to inherit 2 recessive alleles. Unless, of course, we're talking about non-Mendelian genetics. If you remember, there are variations on Mendel's genetics. This simple dominant recessive inheritance is going to be Mendelian genetics. Non-Mendelian genetics includes things like pleiotropy or polygenic inheritance, but I wanted to talk specifically about codominance and incomplete dominance. Codominance, remember, is when you have 2 dominant alleles possible, and if both of them are inherited, they are equally expressed. So if you remember, I am an AB blood type, which means that I am dominant for A and B blood types. My husband is O blood type, which means he is homozygous recessive. So I did a Punnett square here of myself and my husband. And if we were to have children, we could not have any children with an O blood type, because all of my children would inherit either a dominant A or a dominant B. They wouldn't be AB because my husband can only donate the I, the recessive allele. That he cannot donate a dominant allele because he doesn't have one. This would be too, like if we had these purple and white flowers, right? If instead of getting all purple in that first generation, I got purple and white striped flowers, that would be an example of non-Mendelian genetics in that co-dominant inheritance. Incomplete dominance is when neither allele masks the other completely. The most classic example of this being when you cross red and white flowers, you get pink, or if we were to cross these purple and white flowers and we got lighter purple, purple is not completely covering the white, and so we end up with a slightly different genotypic and phenotypic ratio here, because you can physically tell the heterozygous individuals, they will be pink. So if I cross 2 heterozygous individuals, 2 pink flowers, I would expect expect a potential ratio of 1 red flower, 2 pink flowers, and 1 white flower for their phenotypic ratio, which is still that genotypic ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive individual. You will be asked to read a pedigree on the final exam. If you remember, individuals that are filled in have the trait. Individuals who are blank do not. Squares are male. Let's draw a little male symbol here. Circles are female. This is generation 1, generation 2, and generation 3. Lines denote relationship. Uh, Horizontal lines are marriages or partnerships. This kind of vertical line to a horizontal line illustrates children if you have vertical lines coming directly down. So in this pedigree, individuals 1, 1, and 1, 2 are parents to individuals 2, 2, 2, 3, and 2, 5, with individuals 2, 1, 2, 4, and 2, 6 marrying into the family. We see that this particular pedigree, individual 1, 2, has children 2, 3, and 2, 5 that both inherit the trait. And then they have children that inherit the trait, which means that this is a dominant autosomal trait because males and females are equally represented. If we were to skip generations, it would be recessive, or if it's mostly males affected, it would be recessive sex-linked because males inherit one X, and if they inherit the recessive X allele, that would be sex-linked. Again, I went over this in detail in Unit 3, so please make sure to review that. For Unit 4, okay, this is where we talked about protein synthesis, the evidence of evolution, microevolution, and macroevolution. So I think we did really good on Unit 4, if I'm remembering. I think everybody did well on this one. That's great. Unit 4 is going to cover that central dogma of biology. This is the process that makes proteins. So DNA to RNA to proteins is your genotype to phenotype. You will see a connection question about this on the exam as well, probably because you saw one on the Unit 4 exam where I asked you to relate genetics to evolution using at least 4 vocab words or 5 vocab words. Be prepared to do so again, maybe in a slightly different manner, maybe within a slightly different concept, but in that same vein. Remember, the central dogma of biology is a core concept because it is conserved. We see genetic redundancy. We share amino acid sequences. Those codons code for the same amino acids, regardless of which organisms we are talking about, because they are so essential to the actual development of those proteins that they have to be kept conserved, and we have that redundancy built in. Again, no tables on the exam. It is okay for the cheat sheet, but try not to put these complete sentences, put those keywords, put those things on there in shorter answers. If you copy this table directly on your cheat sheet, that's not appropriate. Transcription comes first. That occurs in the nucleus. And so if you see anything related to DNA, the nucleus, RNA polymerase, or the building of mRNA using nucleotides, you are looking at transcription. That is DNA to RNA, and our end product is that messenger RNA, which carries the genetic information through translation. Translation is RNA to protein. This is going to occur in the cytoplasm or on the rough ER at a ribosome. This is going to be the end product to produce protein, and we utilize all three RNAs here: ribosomal RNA, transfer RNA, and messenger RNA to build a polypeptide of amino acids through dehydration synthesis. Keep in mind that you will likely see some sort of matching on this. Do not put these straight up on the test. Keywords are a good thing to add to a table, so maybe we would change this out to the keywords and only put the bold parts on a cheat sheet. This is important to understand because, again, this is connecting a lot of what we learned in the beginning half of the semester in Units 1 through 3 to what we go through for the rest of the semester in Units 4, 5, and 6. That central dogma of biology, remember, is where we have the genotype of an organism being expressed as the phenotype. This is more of what you might see as that connection question. So this is an example of what I would answer You can answer this in any number of ways. It is answered every time I answer it. I do it slightly differently. But if I'm asking about the connection of protein synthesis to genetics and evolution, I am going to definitely be talking about the expression of genotype as phenotype. And if those phenotypes increase the fitness of an individual, natural selection is going to occur, resulting in a higher reproductive output of individuals who are best adapted to their environment. That is going to change the amount of those alleles in the gene pool, therefore leading to evolution of the population over time. That can lead to survival of a species or extinction if it is not beneficial. We might see it in things such as climate change or human impacts on the ecosystem. So this is a big concept that connects what we learned early into what we learn later. Evidence of evolution, I think you guys mostly got this down pretty clearly. We have biogeographical, closer they are to one another, the more likely they are to be related. Fossils, we know they're incomplete, but fossils give us a good picture of relationships. Embryological data, remember this is directly how we identify something as a chordate. It has to have certain traits embryologically in order to be considered a chordate. Those being your dorsal hollow nerve cord, your notochord, your pharyngeal slits, and your postanal tail. Molecular, a very big part of this now. And again, this goes back to that genotype and phenotype and to genetics and into inheritance. So our DNA, RNA, proteins, we talk about how ATP synthase is utilized by both photosynthesis and cellular respiration to generate ATP. That illustrates or demonstrates a evolutionary relationship and the importance of that protein. And then we talked a lot about anatomical traits, those physical similarities through vestigial traits, homologous traits, and analogous traits, which again play a really big role in identifying our diversity of life and our main characteristics of Unit 5. Let's talk about the mechanisms of evolution. If you remember, we have mutation. Without mutation, we do not have evolution. Mutations can be good, bad, or neutral. They can just be what they are. We see them spontaneously arise through proofreading and editing errors or putting in the wrong amino acid, substitutions, mutations, substitutions, insertions, deletions. That's okay, right? Without a change in the DNA, we do not have changes in individual fitness. We do not have changes in individual phenotypes and genotypes. Gene flow is the movement of individuals from one population to another, and it can reduce or increase evolutionary changes depending. Genetic drift is by chance. Everyone is equally susceptible to those risks. And then natural selection is a pressure, and that includes sexual selection where the pressure is the choice of mates. But if we talk about, like, I love this little lizard picture here. This chameleon sees and eats the green bugs better than the brown bugs, and so we see more brown bugs in the population over time. I like that this illustrates this with the same critters in all 4 different things, because these things do overlap. As I think we've hopefully learned in biology, everything overlaps, everything is interconnected. This is microevolution, or your mechanisms of evolution. And then we can have macroevolution, or leading to speciation. And if you remember, we talked a lot about these reproductive barriers, with prezygotic being before baby, before a zygote when the sperm and egg come together, or postzygotic when the sperm and egg have formed. Hybrids reduce fitness. Hybrids are not ideal. Hybrids reduce fitness. They dilute the gene pool. They might result in individuals that are less adapted to the challenges that they face in their environment. And so barriers prevent hybrids, and barriers increase fitness, and barriers then can lead to speciation. Multiple barriers are best because if one fails, another will be there as a fail-safe. Okay, gametic isolation isn't in this image, and so I have a note that gametic isolation is when the gametes don't recognize one another. We talked about temporal, when the time doesn't work, ecological, when they're in different environments, behavioral, when they don't recognize each other as mates, mechanical, when the parts don't fit, and gametic, when the gametes don't recognize one another. Those are all of our prezygotic isolating mechanisms or prezygotic reproductive barriers. Postzygotic is hybrid inviability. The babies die. If your babies die, you have no fitness. Hybrids reduce fitness, okay? Inviability or increased mortality. Basically, the babies die, okay? Hybrid infertility or hybrid sterility is when those hybrids cannot produce themselves. They don't have the ability to produce gametes. Okay. And then hybrid breakdown is when the first generation seems like it's going to be great, but future generations are not viable and therefore reduce future fitness of those individuals. Okay. Um, we did talk about cladograms in both Unit 4 and Unit 5, so I threw it in here, but don't forget that you might see something about it on Unit 5, like you did with the chicken and the egg. Remember, cladograms, the closer we are to the end, okay, I like to think I've been talking about it like a tree this semester. So if this is my tree trunk, right, here's my tree trunk with my other branches, there's my tree. This is the branch coming off of the tree. This is the oldest, this is the newest, because this is where I'm going to see the leaves. And I think if we all imagine trees, that makes sense to us. As such, remember that the traits that are at the closest part to the tree trunk, at the lowest part of that cladogram, are going to be the most ancestral. Everything that branches off after that shares that trait. So sharks, bullfrogs, kangaroos, and humans all have vertebra. They all have a backbone. After the sharks, we see tetrapods, 2 pairs of limbs. Bullfrogs, kangaroos, and humans all have 2 pairs of limbs. Mammary glands are not present in bullfrogs because those are amphibians, but they are present in kangaroos and humans. And then humans are different from kangaroos because we are placental mammals as compared to the marsupial mammals. We look then, and if I say, okay, who is closer related, kangaroos and humans or kangaroos and sharks? We should see that it's kangaroos and humans because they're at the furthest part of that branch. But in addition, we need to say why. We can't just say because they're at the furthest part of the branch. We need to say that they share the most traits in common. Kangaroos and humans have mammary glands, 2 pairs of limbs, and vertebrae in common, whereas kangaroos and sharks only share the vertebrae or 1 trait. That is going to be how we identify ancestral relationships based on that evidence of evolution, like the homologous traits, the analogous traits, the vestigial traits, molecular DNA, embryological, whatever it may be. Cladograms are a visual representation of our evidence of evolution and also help us determine evolutionary relationships within our diversity of life, which is Unit 5. Unit 5, we did chapter 17, 18, and 19, microorganisms being viruses, bacteria, archaea, and protists. Chapter 18 being plants and fungi, and chapter 19 being animals. You cannot use your previously completed tables on the cheat sheet. You will be identifying organisms by key traits and unique aspects. Review the matching from the Unit 5 exam. You will see similar matching on the final exam. Let's go through some basics, though, of what we see with these critters. This is something that I did in the Unit 5 exam review. So viruses, remember, are non-living. They don't replicate. They don't have a cell. Or they replicate, they don't reproduce, they don't have a cell, they don't have those key characteristics of life. You need all 6 to be considered alive. Viruses do have that lytic and lysogenic cycle, and they have a protein capsid with genetic information. Remember, we cannot use antibiotics to treat viral infections because they do not have the components that antibiotics target. In doing so, we increase the antibiotic resistance in bacteria that exist in your body by putting them under that selection pressure to survive antibiotic treatment. Bacteria are prokaryotes. They are unicellular with a unique peptidoglycan cell wall. They have unique shapes in the sphere, the rod, and the spiral. And that is something that we see there. They're everywhere. They eat everything. They do binary fission as a reproduction. They do a form of mitosis. Archaea are also prokaryotes. So when I asked you on the Unit 5 exam to compare bacteria, archaea, and protists, what you need to know to identify them, it's not just that bacteria are unicellular, because all 3 of these are unicellular. Bacteria, archaea, and protists are all unicellular. Bacteria are prokaryotes, but so are archaea. So we have to add in those unique traits, those key traits, the things that are unique to that group. Bacteria, that would be the peptidoglycan cell wall. Archaea, it would be the unique lipid bilayer. Protista is that it is a eukaryote, which does not allow it to be an archaea or bacteria, but it is not a plant, animal, or fungus. So hopefully you clarified that for yourselves on the exam. Keep that in mind. That might be something that I would put on the chart or on a cheat sheet. I would probably include for my cheat sheet this domain kingdom example. I would put that on there. I would put the unique traits. I suppose I should have just bolded this instead of highlighted it. Give me a second. I want to make that change here. Bold, remove the highlight, and then an example. I wouldn't necessarily put their reproduction style. I wouldn't necessarily put their cell type unless you can't remember that. That might be a good thing to add in there, but you could combine that into unique traits. You don't need to worry too much about nutrition and reproduction. So if we look at this, then we're saying, okay, can I tell the difference between these major classifications of life that we went through? You should be able to tell me that a fungi is a eukaryote, that there are unicellular yeast and multicellular mycelia. Plants are all multicellular. It's why plants are a plant and algae is a protist. Animals are all multicellular. We know that animals have muscles and nerves for movement. We know plants have roots and shoots. We know that fungi have chitin in their cell walls, which is analogous to the cell walls in plants that have cellulose. We know that protists is a catch-all group and they don't really fit. Archaea are extremophiles, okay? This should be fairly fresh in our minds, and I don't go through all the little details here because if I were doing a cheat sheet, these the 4 sections would be what I would put on my cheat sheet. You should hopefully be able to recognize animals by their classifications, mammals being like animals with fur and mammary glands, or insects having flight, and an exoskeleton. Definitely review that part of the cheat sheet. We do know that, if we find something in an extreme environment with a unique lipid bilayer that's a unicellular prokaryote, it's archaea. If we find something in an extreme environment with a peptidoglycan cell wall and it's a unicellular prokaryote, it's bacteria. We do find bacteria in extreme environments as well because they are literally everywhere. But archaea are what we can call our extreme loving. It's just how we tend to kind of specify the difference. But again, that big difference is between the lipid bilayer and the peptidoglycan cell wall, and the fact that archaea share ribosomes with eukaryotes. Protists are a catch-all. Fungi they have the chitin cell wall. Plants have a cellulose cell wall. Animals do not have cell walls. Okay? And then last but not least, Unit 6. So Unit 6 is levels of ecology, population ecology in Chapter 30. Biodiversity, including community interactions in food webs, keystone species, and biogeochemical cycles in 31, and the biosphere and human impacts in 32. This one we tend to feel very comfortable with because it is material that is common in nature documentaries. But just a quick reminder, levels of ecology is least to most comprehensive, with the smallest being organismal, then population. Organisms make up populations. Populations make up communities. Communities are in ecosystems with the abiotic factors, and then the biome is the whole world. Remember, this ties into those characteristics of life. We have the biotic versus abiotic components, biotic meaning living, abiotic meaning non-living. And remember, we need to know what makes something alive, which ties back to those 6 characteristics of life. That is how we determine if in an ecosystem something is abiotic factor or a biotic factor. And if you do remember, the populations are often regulated by density-dependent and density-independent factors, which are generally quantified as biotic factors for density-dependent or abiotic factors for density-independent. This also ties into the community and ecosystem interactions. This is one of those ones that, again, we have a lot of gray areas and overlap. Remember, predation is going to determine population sizes, food availability, competition, disease, and then non-living factors that can affect organisms are absolutely things like temperature, light intensity, water, pH, gas. We've talked a lot about how if the ocean dies, we die, and that would happen because of the ocean warming and becoming more acidic and resulting in a loss of those producers, which again would reduce the amount of oxygen in the environment and the amount of food available to us as consumers. That's an abiotic factor. It doesn't matter how many individuals are here, it's going to have an impact on those living organisms. We did talk about the community interactions as well as biodiversity. A good biodiverse community is going to be evolving together, and we're going to see balanced community interactions. We did talk about mutualism, commensalism, consumption, and then competition. And remember, we talked about it in terms of, plus, 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 minus, plus, zero. I have a couple more on here just because I think it's of interest. But parasitism, predation, and herbivory are all consumption, which is a plus minus, and that's going to reduce future fitness. Remember, these pluses and minuses are in terms of energy, which is in terms of then fitness and usually in future fitness. Commensalism is a plus zero, where neither one individual benefits and the other one is not affected. And then competition is a minus minus, where both individuals lose future fitness. And mutualism is a plus plus, where individuals benefit from their interactions. I did also throw that biodiversity on here in terms of community A and community B. Remember, when we look at biodiversity, we're looking at the species richness, so how many species are present. In this case, we have 1, 2, 3, and 4 species present in both environments, and they have the same species present. So species richness is equal. 4 species in both, same species in both. But what I hope we notice is that in community B, we see that the grasses dominate. And so we actually only have 1 of these little yellow flowers and 2 of these little red flowers and only 1 tree, which is going to result in a lower evenness in B, which means that a lower diversity of B. Okay. We want to see a good species richness and a good species richness and a good species evenness. I actually had that on here twice, so we're going to remove that. Food chains versus food webs. Food chains are a good way to see some basic interactions, but it is not enough because it does not show the full picture of community structures. Food chains, remember, if you are a producer, you are going to be the base of a food chain. Producers are consumed by primary consumers or herbivores like grasshoppers. Herbivores are consumed by secondary consumers like frogs. So primary to secondary, we are 2 steps away from the grass. Tertiary consumers are going to eat secondary consumers who eat primary consumers, and then quaternary consumers, this should say quaternary consumer here, let's change that, so 4 prime, are going to be our top level consumer that eats the tertiary, which ate the secondary, which ate the primary. Remember, we only have 10% of energy moving through each part of these, and that's that rule of 10. And nobody gets away from decomposers, which do actually feed back into our producers. When we look at a food web, it's a lot more complicated than that. If we go back to the grass, let's just look at the grass here. Again, this is our producer. Grasses are consumed by the grasshopper, who are primary, who's consumed by the shrew, which is secondary, who's consumed by the hawk, which is tertiary. If the grasshopper is consumed by the bird is secondary. If the bird is eaten by the hawk is still tertiary. Okay. If the shrew is consumed by the snake is tertiary because the shrew ate the grasshopper. The shrew is then eaten by the snake. So we are 1, 2, 3 steps away. And then if that is eaten by the mountain lion, that makes that quaternary. But a mountain lion might eat a shrew, which also makes the mountain lion tertiary in this system. So this would be a tertiary because 1, 2, 3 steps away from the producer. 1, 2, 3, 4 steps away from the producer. If they look at the grass, it's eaten by the rabbit as well, which makes the rabbit primary. If the mountain lion eats the rabbit, that means the mountain lion is secondary, tertiary, and a quaternary consumer because it is eating at all different levels in that trophic level. And again, that's just looking at the grass. When we look at everybody else, it gets to be a little more complex than that. Quick reminder, keystone species increase biodiversity when present. Invasive species decrease biodiversity when present because invasive species are moved into the environment intentionally or unintentionally and compete with native species. I like these 2 images a lot because it does really illustrate this. So when we have a sea star in a tidal pool, sea stars actually eat a lot of different filter feeders or carnivorous individuals. They eat a lot of filter feeders. When they're eating those filter feeders, that allows the algae to grow, the nudibranchs, the sponges, everybody is able to live, and we see a balanced ecosystem. And so these arrows are pointing the other direction because it's also showing this like direct relationship here. When we remove the starfish, which was actually the research that was done first to determine a keystone species, we see that we lose biodiversity. Mussels become very abundant. They are filter feeders. They eat a lot of the algae. That then results in fewer barnacles, fewer epiphytes, no sponges, no nudibranchs. We lose the anemones. We lose the tunicates. Like these guys, these mussels just outcompete everyone else. The sea stars are going to keep those populations in check. That is the key with a keystone species. Invasive species decrease biodiversity. So this image is showing the same sort of thing, but instead of showing like a reduction in the population, they show like a smaller image. So for instance, Norway rats were introduced on islands. These rats eat baby birds. If you eat the babies, they don't get to be adults. It decreases the fitness. When the birds are no longer present in the environment, your chitons and snails increase in population abundance, which then results in a decrease of the algae because they overconsume that algae. Again, this is one of those density dependent factors directly related to other species. If we don't have the Norway rats, the gulls and oyster catchers, these birds that are here, do eat a lot of these mollusks, which allows for an increase in the algae, which you can imagine then has a big increase on the population as a whole, because again, without producers, we lose consumers. Okay. As last slide here, and our last like 5 minutes, look at this. I said an hour, 15 minutes. I'm going to get there. Remember that the biogeochemical cycles tie back to that chemistry of life. The nitrogen cycle is incredibly important. We are going to see the nitrogen cycle as a way of maintaining a balance. We see a lot of the biotic community. Nitrogen is really important for proteins and nucleic acids. Cho-cho-chan-chan, proteins and nucleic acids both require nitrogen. So again, without that good solid nitrogen cycle, we lose out on some of those major macromolecules. And as we know, without DNA, you can't do mitosis, you can't do meiosis. Without proteins, you can't do any of those things, and so nitrogen is really important. Humans are disrupting the nitrogen cycle through use of fertilizers and overabundant reliance on fossil fuels. There are ways to go around that. There are positive things that humans do, but we have to be sure that if we're utilizing things like fertilizers, we are doing so appropriately. The phosphorus cycle is also really important, again, for those nucleic acids. No phosphorus, no ATP, because that's adenosine triphosphate. No RNA, no DNA. That means no protein synthesis, no life, right? No phosphorus, no life. But phosphorus is generally biologically kept. It is going to be consumed and produced very little through those decomposers as well as through your plant materials. It can be released through weathering of rock. That is a slow process. We are disrupting it again with fertilizers, and hopefully you remember some of those videos I showed of nutrient pollution in the waterways. Carbon dioxide in the atmosphere is generally balanced in biotic communities. Carbon is the backbone of all of our macromolecules, and as such, it is relatively kept by living organisms and transferred between producers and consumers and decomposers. We are unfortunately disrupting that with the burning of forests, the combustion of fossil fuels, the removal of forests, which removes our photosynthesizers, which are carbon fixers. We're increasing the amount of carbon in the oceans, which is leading to acidification, which again reduces the life of the oceans. And, it all is a cascade effect. We are part of our environment. We are part of our ecosystems. There are good things you can do: reduce, reuse, recycle, vote. Doing things in your own environment and community. It's not all bad, but remember, humans are having huge impacts on the biosphere as a result of overuse of land, habitat degradation, pollution, all of those things we talked about in chapter 32. I'm sure they all stuck with you because it does get a little heartbreaking. But remember, there are good things we can do. River restoration, removing of dams, removing invasive species in your yard. Even that little bit counts. Even that little bit matters. So that is our very quick review for both the Gen Ed assessment and the final exam. There is always more. Please make sure you review your exams. Please make sure you review those past unit review videos that I have posted. Look through the PowerPoints. I know that they're not as, the review PowerPoints aren't as complete, comprehensive. Work on your cheat sheets. Let me know if you have questions. Have a wonderful day. Take this Gen Ed assessment very seriously, please. It is something that is required by the powers that be, and it is something that does reflect on our course as a whole. I want to see what you've learned. I know you've learned. You guys have shown me this all semester. This is just one other way for it to see it, and it will also help you develop your cheat sheet because it'll tell you what you forget or what you remember. I think that's it. Thank you guys. Good luck on your Gen Ed. Have a good day.